morning. It's very nice to see all of you here today. So I intentionally repeated the words 25 years in my title to indicate that in vivo research or animal research is something that ALS TDI has been doing since day one. That's what we built our reputation on. However, when I showed my presentation to Ken Thompson, he corrected me and he reassured me that no animal research was happening at the Haywood basement. I'm sure you've seen all this photo. In vivo research started a year later when TDI made its first lab at the New England School of Medicine. It's important to know what the framework, what we knew about ALS at the time in order to realize the ALS TDI approach. So in 1999, the SOD1 gene was the only confirmed ALS gene. There was another one starting to emerge, but essentially SOD1 was the only one. And the SOD1 mouse model was the only genetic model available. The molecular mechanisms of ALS were not known. The heterogeneity of the ALS was not known, and there were no biomarkers for ALS. So the staff at ALS TDI, Fernando was there, Ken was there, many of the people that are still today did what was reasonable at the time, and plus the urgency of trying to save Steve Haywood, who was still alive. So they took whatever drugs were reported as beneficial by other patients or some drugs repurposed from other indications, and they tested them in the animal model that was available, simply asking whether the mouse improves, if it lives better. And they did this over and over and over again. And as a result, they became very good at running this animal model. This is something that we're known for to this day, and many achievements were reached. This is not to be a comprehensive list, but I listed some of the main achievements of ALS TDI over the years. The early years were mostly learning how to use this mouse model, developing techniques, developing NeuroScore, which is the method we use to measure disease progression. And then in 2008 came the seminal paper that listed the optimal design of mouse studies. This has been adopted since by labs all over the world. And around the same time, we developed a transcriptome profile of SOD1 gene, of SOD1 mouse, meaning gene expression in all its several tissues, and we generated a database that we still use today. And soon after that came the discovery of MR1, which was published in the Nature Genetics paper. Last time I measured the number of studies we have done was over 500 survival efficacy studies. These are long 3 or 4 month long studies, each one with several mouse models, testing several 500 therapeutics. Now there's probably more. And we tested thousands other therapeutics in smaller studies. And all this progress culminated into tegoprubart, which was tested in a phase 2 clinical trial in 2022 by Eledon Pharmaceuticals. Since then, we have learned a lot more about ALS. We now know that more than 40 genes are linked to ALS. Several genetic ALS animal models have been developed, many of them with caveats. We have characterized several of them, over 20 of them at ALS TDI. The cellular mechanism of the disease are better known, not completely, of course, but now we have a lot of biomarkers that we can use both in humans and in our animal models. And our job as in vivo pharmacologists is to back translate the findings from humans into animals in order to develop more accurate models of ALS and readouts that would translate better into the clinic. The etiology of ALS might be complex, but there are some pathologies that are central to all types of ALS, sporadic or familial. Fernando mentioned about the motor neuron pathology, which you can see in all ALS patients. On the left, you can see a normal motor neuron receiving inputs from other neurons, sending electrical signals through its axon to terminals. Through the neuromuscular junction, it transmits information into the muscle, initiating movement. This process goes wrong in ALS long before you might even see a motor neuron death. The axons often get sick and they release their cytoskeletal component neurofilament into the bloodstream. You'll hear a lot about neurofilaments today that we can measure. Because as the axons decline, the electrical activity of the muscle declines. We can also measure that nowadays in animals and humans using a technique called CMAP. ALS can be quite heterogeneous, but many features are common to all patients, like muscle weakness or paralysis or respiratory problems. I say all this to mention that the models, the animal models that capture many of these cardinal features of ALS that are common to all patients will better translate into the clinic. This is not to say that one model will be good for everybody, will represent all ALS patients, but the more of these common features a model has, the better the model. We now have 3 mouse models in ALS in which we are actively testing drugs. These are models that have many of those features that I previously mentioned. They're all based on ALS-linked mutations. I'm talking about the SOD1 G93A mutation, the profilin-1 G118V mutation, and the TDP43 Q331K mutation. 2 of them have a decreased survival. They all have motor deficits. They all have neurofilament elevations. They all have CMAP depletions, but they all have different disease progressions, similarly to what happens in the human population. We also have a zebrafish, C9orf72 animal model that also shows decreased survival and motor deficits, and I'll talk about it towards the end of my presentation. I will now describe how NFL elevations and CMAP depletions, the side effects of motor neuron death that I mentioned earlier, compare in these 3 animal models. So neurofilaments are cytoskeletal components of the axon. They're considered a validated biomarker of ALS these days, and they're included in clinical trials. And tofersen was actually approved based on its ability to decrease NFL in SOD1 patients. Thanks to the efforts of Andy Moreno, who is very good at drawing blood from animal models, we can now analyze NFLs longitudinally in the mouse models, meaning from the same animal over and over again. And we can measure it in plasma using one of 2 supersensitive immunoassays called CMOA and MSD platforms. I show you here the longitudinal changes in our 3 animal models as they age. The SOD1, the profilin-1, and the TDP43 one. The first thing you'll notice is that the transgenic animals, the ones with the open circles, have higher neurofilament levels than the wild-type controls. The axis in all three panels, the x-axis is the age, and then on the y-axis is the amount of neurofilaments. The second thing I'd like you to notice is that the amount of neurofilaments is quite different. If you notice that the y-axis, they all differ by a 0, meaning there's an orders of magnitude more neurofilaments in the blood of SOD1 mice compared to profilin mice, compared to TDP-43 mice. And this roughly, this correlates with the disease progression. The most aggressive models have more neurofilaments in the blood than the least aggressive models. And finally, I'd like you to notice that in the SOD1 and the profilin-1 model, the neurofilament levels go higher as they age. They reach their most levels just before the mice die, while in the case of TDP-43, where there's no decreased survival, the NFL levels are consistently higher than controls, but they bounce around a lot. We wanted to see whether neurofilament levels are predictive of disease progression in the animal models. For this reason, we correlated the levels of neurofilament at early stages, long before the mice present with any symptoms. And then we looked at when the mice actually died, and we found perfect correlation in both the SOD1 and the profilin-1 model. If you look at both of these graphs, the mice that had a lot of neurofilaments died earlier, and the mice that did not have so many neurofilaments in their bloodstream died later. Traditionally, we've been measuring neurofilament levels using the CMOA assay at Quanterix, but we made, thanks to the efforts of Kaylee Miller, we made the strategic decision to bring this method of measuring neurofilaments in-house using the MSD platform. And we were successful at doing that. So we now measure neurofilaments at TDI. The absolute NFL levels differ between the two methods, but they correlate very well and they convey the same information essentially. This is important because now we can do our studies quicker. We have a quicker turnaround time. They're cheaper and they're more scalable. We can do a lot more samples. The other technique that was initially developed in humans, and now we use it in mice to measure the progression of the disease, is called compound muscle action potential, or CMAP, which we brought in about 5 years ago. Briefly, we anesthetize the mice. We insert a small stimulating electrode to initiate an action potential, and then we measure electricity or potential, as you say, in the downstream muscle. The amplitude is higher as when the mice are young and healthy, and as the disease progresses, it decreases. I'm showing you here longitudinal CMAP changes in our 3 models. You will notice that it's almost the reverse of what I showed earlier with NFL levels. So NFLs increase and CMAP decreases. In the SOD1 mice, there's differences, the significant difference in the CMAP, or the electrical activity of the muscle, as early as 50 days. This is before we see any symptoms in mice. We don't see symptoms until age day 90 or 100. So we can detect differences early, and then they decrease from there. In profilin mice, there are significant differences in CMAP, but they mostly happen later on in the disease course, and then they progress. In the TDP-43 mice, there are changes early, but like in the case of neurofilaments, they remain stable. They don't decrease further. Similarly to neurofilaments, CMAP levels at a very young age are very predictive of disease progression in both SOD1 and Profilin-1 mice. I'm not showing you the TDP-43 mice because obviously it doesn't progress. So mice that had low CMAP values present with the disease sooner and they die earlier, and those that have high CMAP values, those with more functional muscles, live longer. Another technique that we brought in the lab in the last year, due to the efforts of Josh Kidd and Matt Ferrola, is known as whole body plethysmography. So breathing abnormalities are common to people living with ALS. This is something that we never measured in mice before. So we decided to bring this technique where we put mice in a tightly sealed container. We measure temperature, humidity, and we apply a steady level of airflow. And then using a pressure device, we measure the pressure, which is thanks to the respiration of the mouse. And we get graphs similar to the ones that you see here. On the top is a mouse that breathes normally, while at the bottom is a mouse that has respiratory issues. And we have already tested it in the SOD1 mice, and we see respiration changes at age day 90, like inspiration time and inspiration and expiration flow. The amount of air that they breathe over a minute and their breathing frequency is significantly declined in the SOD1 mice compared to controls. We're now in the process of determining respiration parameters in the other two models. How about the fish? I mentioned them in the very beginning. I want to make a disclaimer that I have worked with mice for many years. If I say mice instead of fish, I apologize. So Val Tassinari is leading the fish program. She has in her possession now 5 different models of zebrafish. Some of them are transgenic, but some of them are wild-type zebrafish. They all model ALS in humans. So she either exogenously applies dipeptide repeats, which are ALS-causing proteins in humans, or the transgenic models endogenously produce those. I should say that the transgenic zebrafish were donated to us by Tenor Ramesh, who was part of ALS TDI 25 years ago. So talk about a complete circle. They all show decreased survival, except a control transgenic line at the end. And they all show motor deficits like the mice do, in this case, swimming decline. A couple of assays that Val uses in the zebrafish, just to show you how zebrafish research works. In this case, this is using wild-type zebrafish, very young, 4 days post-fertilization, the larval stage, and she applies different concentrations of PR15 and GR15. These are the dipeptide repeats that cause ALS. And then she observed that PR15 causes a dose-dependent decrease in survival in zebrafish, while GR15 does not. And her efforts right now are focused on determining why this is so. We can also have behavioral assays measure the locomotion using an instrument called ZebraBox that measures the speed and the distance traveled of young zebrafish in a multi-well plate format. So each well contains one fish, and Zebra Box measures, you can see the tracking lines, how much they travel, how fast they travel. And we're trying to characterize all the transgenic lines right now for their ability to move and survive, and eventually to screen drugs in this assay to see whether they can make a slow zebrafish move faster and so forth. So the main takeaway, what I try to convey today is that as we learn more from ALS, from humans, our ability to better model ALS in animals and more effectively test therapeutic improves. The quality of our mouse model will directly translate into better drugs for humans. In the future, we hope to expand and diversify further our animal model portfolio. We no longer think that one model can cover all types of ALS, so we have to have multiple models. For example, a good one to have would be a C9orf72 based model for mouse model. We will also like to have more predictive biomarkers like NFL, especially blood-based ones that we can measure longitudinally, as we learn more from human studies, perhaps from our own ARC study. A good one to have would be a plasma-based inflammatory biomarker. Inflammation is a part of ALS. And finally, we'd like to optimize the quality of our preclinical candidates for in vivo testing, for example, by leveraging the discoveries of our own cell biology team or through collaborations. I presented many of them last year. I'd like to acknowledge all the current members of ALS TDI in vivo team whose work I presented during my talk, but also the work of all the past members of ALS TDI. Thank you, and I'd be happy to take any questions.
Hey, Theo, the people at home can't hear the floor mic, so can you just repeat the question into your mic when we get questions?
Your mic?
Hey, Theo.
Yeah. Okay.
Okay, so my question is about the predictive nature of the neurofilaments. Is what you're looking at now a measurement of the predictive value of the rate of progression, or is it the predictive nature of the disease itself? Like if someone had blood work done and they have no symptoms whatsoever, but this biomarker showed up, is it saying like, okay, the likelihood of you having ALS is pretty high here?
Mm-hmm. Thank you. The question has to do with the predictive nature of neurofilaments. I will say that I was talking strictly about the animal models, not the human studies. Fernando will talk about human NFL in the afternoon. So it turns out that in mice, early neurofilaments predicts disease progression, and we hope to use that in order to assess the efficacy of therapeutics. If it does predict disease progression, you would think that a drug that decreases neurofilaments will increase disease progression, and that's how we plan to use it in the future. I'm not sure if I answer your question, but again, this is strictly about animal models. Whether it would predict disease progression in humans remains to be seen. It's more, my impression is more of a binary thing. High levels would signify the presence of ALS or other neurological diseases. It's not strictly about ALS neurofilaments.
And that's all we're going to have time for.
That's all we have. Thank you very much.